Hello, my name is Dr. Christopher Turner. I'm a research fellow at Monash University and adjunct research fellow at the University of South Australia. However, this study was conducted when I was a postdoctoral researcher in the Granville Lab at the University of British Columbia in Vancouver, Canada. Emerging evidence has identified that Granzyme K, a serine protease typically expressed at low levels in healthy tissues, is elevated in response to inflammation and has pro-inflammatory properties. As atopic dermatitis is characterized by inflammatory eczematous lesions, we hypothesized Granzyme K to be elevated in atopic dermatitis, thereby contributing to both inflammation and disease severity. So how did we do this study? Firstly, we evaluated Granzyme K expression in a panel of lesional atopic dermatitis tissue samples, comparing them to patient-matched non-lesional control skin and healthy skin controls. Next, to determine a pathological role for Granzyme K in dermatitis, like skin inflammation, dermatitis was induced and assessed in wild-type and Granzyme K knockout mice. We had several main findings. Granzyme K was elevated in atopic dermatitis patient lesions compared to healthy skin controls. In mice with dermatitis-like skin lesions, Granzyme K knockout led to reduced disease severity, including smaller lesions and reduced scaling. Surprisingly, absence of Granzyme K did not reduce inflammation. Rather, Granzyme K knockout mice exhibited improved angiogenesis and vasculature, as well as decreased microvascular damage and microhemorrhaging. Granzyme K also activated PAR2, a mediator of itch, increased expression of angiogenic factor VEGF, and enhanced skin fibrosis. This study is relevant to dermatologists and patients. We identified Granzyme K to mediate several mechanisms important in inflammatory skin disease pathogenesis, namely vascular dysfunction, itch, and fibrosis. As such, Granzyme K is a potential therapeutic target for skin conditions associated with persistent inflammation, itch, vasculitis, and pathologic angiogenesis.